Hello, I am a third year medical student from BPKHS Dharan. In this video, I am describing about the physiology of growth hormone, that is the role of growth hormone in growth and metabolism and its regulation. In intrauterine life, before birth, many hormones they play a role in growth, like growth hormone, insulin-like growth factors, thyroid hormones, glucocorticoids, androgens and estrogens. But after birth, that is in post-uterine life, only growth hormone and insulin-like growth factor 1 play a vital role for growth. So what are the abnormalities if there is excess or deficiency of growth hormone? It depends upon the time of life, that is on the age. If growth hormone is excess before puberty, that is before the closure of the epiphysis with the shaft of the bone, then there will be gigantism, there is increased linear growth. And if there is growth hormone deficiency in childhood, then it results in pituitary dwarfism. And if growth hormone is excess in adult, that is after puberty, then there will be acromegaly. And growth hormone deficiency in adults does not cause serious clinical illness, but it has been found that if growth hormone is replaced from outside, then it results in increased lean body mass, decreased body fat, and gives a sense of well-being to the patient. In addition to its role in growth, growth hormone plays an important role in metabolism. Now we will see the effects of growth hormone on protein, then on carbohydrate, and then on lipid metabolism. Let's first understand the process of synthesis of proteins. In nucleus, mRNA is formed from DNA. This process is called transcription. mRNA so formed in the nucleus is diffused into the cytoplasm, and this mRNA in cytoplasm is translated into proteins. This is called RNA translation. Ribosomes, they synthesize proteins from amino acids. Proteins so synthesized, they are broken down into amino acids for the fulfillment of the energy requirement of the cells. That is protein catabolism. Now we will see the effects of growth hormone on these processes. I have shown by numbers 1, 2, 3 and 4. First, number 1. Growth hormone causes enhancement of amino acids into the interior of the cells. When amino acid concentration inside the cells is increased, then ribosomes they can synthesize more proteins. Number 2 effect of growth hormone is it increases RNA translation. There is more proteins from mRNA. Third effect of growth hormone is on transcription. It enhances the transcription of DNA in the nucleus to form more mRNA. So if there is sufficient concentration of amino acids, sufficient vitamins and sufficient energy inside the cell, then by acting on transcription of DNA, growth hormone can increase the synthesis of proteins. So 1, 2 and 3 effects, they cause more protein synthesis. Now let's talk about number 4. Number 4 effect of growth hormone here is it inhibits the catabolism of proteins. That is, it does not let proteins to be broken down into amino acids. Why is this effect? Is because growth hormone it mobilizes free fatty acids from adipose tissue, and those free fatty acids they undergo oxidation to fulfill the energy requirement of the cells. That's why proteins will be reserved, they will be spared. So the net effect of growth hormone is increased tissue protein deposition in the tissues and cells. Now the role of growth hormone in lipid metabolism. The main action of growth hormone in lipid metabolism is it increases lipolysis. In adipose tissue, fat is stored as triglycerides. This triglycerides is broken down into free fatty acids. This is called lipolysis. These free fatty acids then they are released into blood from the adipose tissue. These free fatty acids they are uptaken into liver and muscles. In the muscles, free fatty acids they undergo beta oxidation to produce acetyl coenzyme A, and this acetyl coenzyme A goes into Krebs cycle to produce ATP, that is, it gives energy to the cells. In the liver, Again, from blood, free fatty acids, so released from the adipose tissue, they are uptaken. Free fatty acids, they undergo beta oxidation in liver, similar to muscles. Acetyl coenzyme, so formed from beta oxidation of free fatty acids, it can undergo two processes in liver. First, it can go into Krebs cycle to produce energy, and next, acetyl coenzyme A, it can produce different ketone bodies like acetoacetate, beta hydroxybutyrate, and ketone. But in muscles, ketogenesis does not take place. So if there is excess growth hormone, as in acromegaly, there can be ketogenic effect, that is, there may be elevated ketone bodies in the blood. Now the effect of growth hormone in carbohydrate metabolism. These effects of growth hormone are mainly because of one fact, that growth hormone causes insulin resistance. The precise mechanism of how growth hormone causes insulin resistance has not been found yet, but it has been hypothesized that since growth hormone, it increases lipolysis, there are increased free fatty acids in the blood. These increased free fatty acids cause insulin resistance. Insulin normally does two major functions. First, in the skeletal and muscle cells, it increases the glucose uptake into the cell. Next, in liver, insulin, it inhibits gluconeogenesis. So now, since there is insulin resistance because of growth hormone, gluconeogenesis will increase to release more glucose from liver into blood, and glucose uptake will be less. So also, because there is insulin resistance, there will be compensatory increase in insulin secretion. So the effects of growth hormone on carbohydrate metabolism can be summarized into 3 points. First, increased gluconeogenesis. Second, decreased glucose uptake. And third, increased insulin secretion. Now the main function of growth hormone is to increase the growth of the skeletal frame, that is bones. Growth hormone, it has 3 important effects on the growth of bones. On the left side is chondrocytic cell that causes the deposition of cartilage, and on the right side is osteogenic cell that causes the deposition of bone. The 3 effects of growth hormone are: first, in both cells, chondrocytic cell and osteogenic cells, growth hormone causes increased protein deposition by the mechanisms which we have previously discussed. Second effect is growth hormone increases the rate of production of both cells, chondrocytic cell and osteogenic cell. Third effect is growth hormone, it can transform chondrocytic cell into osteogenic cells. These 2 schematic diagrams, first and second, they show the mechanisms of bone growth. In the first figure, we can see epiphysis at the end of the bones and shaft at the middle. Between epiphysis and shaft, growth hormone causes the deposition of new cartilage. This new cartilage is then converted into new bone. New bone so formed will again increase the distance between the epiphysis and shaft, and the process continues. So this causes the linear growth. But at the age of puberty or adolescence, epiphyseal cartilages, they will be used up to form bone, and no further linear growth will occur now. Next diagram shows the growth of the bone once the epiphyses have been fused. In the bone periosteum, that is outer layer of bone, in the bone periosteum and in the bone near the marrow, that is near the cavity, there are osteoblasts. These osteoblasts, they cause the deposition of new bone to cause the thickening of bones. In acromegaly, there is thickening of jaws. There is protrusion of the chin and lower teeth, and there is protrusions of the supraorbital ridges. This is because of the second mechanism. Though the epiphyses, they have been fused, there will not be linear growth, but because of the deposition of new bone by osteoblast in periosteum and marrow cavity, there will be thickening of the bones. Before going into the regulation of growth hormone, one important thing to know is growth hormone does not show its effects on its own, but it produces some other mediating factors like insulin-like growth factor 1. Growth hormone, it binds to the growth hormone receptor on its target tissues like liver, muscle, kidneys, and from these target tissues it forms the mediating factors like insulin-like growth factors. Among them, insulin-like growth factor 1, also known as somatomedin C, is major. It is called somatomedin C because it mediates the somatic effects of growth hormone. But most of the insulin-like growth factor 1 is formed from liver. Growth hormone, since it is not bound in much quantity to plasma proteins, its half-life is very low, that is about 20 minutes. But about 90% of the insulin-like growth factor 1 is bound to plasma proteins, so its half-life will be prolonged, that is about 20 hours. In Laron dwarfism, there is mutation in the growth hormone receptor, because of which growth hormone, it cannot bind to growth hormone receptor and cannot produce insulin-like growth factor 1. So in Laron dwarfism, growth hormone is either normal or is elevated, but insulin-like growth factor 1 is decreased. This diagram shows the regulation of growth hormone. Growth hormone, it is produced by the somatotrope cells of anterior pituitary gland. These somatotrophs, they are under the control of hypothalamus. Growth hormone releasing hormone, which is produced from arcuate nucleus. Here blue colored nucleus in hypothalamus, we can see. This nucleus releases growth hormone releasing hormone, and this releasing hormone acts on the somatotrophs to increase the growth hormone secretion. Whereas somatostatin, which is secreted from the nuclei of periventricular region of hypothalamus, here shown by red colored nucleus, it inhibits the somatotrophs to decrease the growth hormone secretion. Now let's see the molecular mechanism of growth hormone releasing hormone and somatostatin on somatotropes. This cell, small cell shown, is the somatotroph. Growth hormone releasing hormone, it acts on the G protein coupled receptor. This receptor is coupled with GS, stimulatory G protein, which is subunit alpha S, beta and gamma. When growth hormone releasing hormone, it binds to this receptor, alpha S will be detached and will bind to adenylyl cyclase, AC here. When adenylyl cyclase is activated, it converts ATP into CAMP, cyclic adenosine monophosphate. CAMP will phosphorylate protein kinase A. Protein kinase A will cause increased influx of calcium into the somatotroph. When intracellular calcium concentration increases, then preformed growth hormone will be released from somatotrophs. Somatostatin, how it inhibits somatotroph? Somatostatin, it binds to G protein coupled receptor, again similar to growth hormone releasing hormone, but it binds to inhibitory G protein. When alpha I subunit of inhibitory G protein binds to adenylyl cyclase, AC, it inhibits it, and ATP will not be converted into CAMP. CAMP will not phosphorylate pKa, protein kinase A, and influx of calcium cannot take place. There will be decreased intracellular calcium concentration, and preformed growth hormone cannot be released from somatotrophs. Now let's see the negative feedback by growth hormone and insulin-like growth factor 1. Growth hormone, it inhibits its own secretion by acting on somatotrophs. This is called short loop feedback. Growth hormone, by acting on target tissues, especially liver, produces insulin-like growth factor 1. Insulin-like growth factor 1, it has got important role in negative feedback suppression of growth hormone. It has been shown by green color lines A, B, C. Number A effect is insulin-like growth factor 1, it stimulates periventricular nucleus to increase the amount of somatostatin, and it indirectly inhibits growth hormone. Next effect of insulin-like growth factor 1 is it inhibits the arcuate nucleus of hypothalamus, because of which growth hormone releasing hormone will be produced in less quantity, and ultimately growth hormone will be decreased. And third effect is direct effect of insulin-like growth factor 1, that is, it can directly inhibit somatotrophs to decrease the growth hormone secretion. In addition to growth hormone releasing hormone and somatostatin, here are other factors that stimulate and that inhibit the growth hormone secretion. On the left side are the factors that stimulate the growth hormone secretion. They are decreased blood glucose, amino acids and free fatty acids in blood. Exercise, it stimulates growth hormone secretion. Likewise, trauma, stress and excitement stimulates growth hormone secretion. Deep sleep, especially stages 2 and 4, stimulate growth hormone secretion. Ghrelin, which is hormone produced by endocrine cells of GI tract, especially fundus of stomach, it also stimulates growth hormone. Ghrelin, it is produced in response to fasting. Likewise, starvation and fasting also will stimulate growth hormone secretion. On the right side are the factors that inhibit growth hormone secretion. If blood glucose and free fatty acids, they are increased, growth hormone will be inhibited. Likewise, with the age, growth hormone secretion will decrease. Obese persons, they will have less growth hormone secretion. And if growth hormone and insulin-like growth factors are given exogenously, then they will also inhibit growth hormone secretion.